So guys, now we get into lipids for a little bit. And when you look at lipids, first thing I want you to notice is that there's no nitrogen there, right? So pay attention to that. There's carbon, hydrogen, oxygen, no nitrogen. It goes on to say that lipids are one of the main sources of energy for the body, okay? And this is the way that this has been asked on an old test. So take a look at this. So it's kind of picky, but greater than 60 minutes is a number you want to pay attention to. Now, as you get into fatty acids, you guys might remember there's saturated and unsaturated fatty acids. Saturated, connect that with the single covalent bonds. So connect the S's there, saturated, single. Whereas you get to unsaturated, it's joined by double covalent bonds. Remember, covalent means shared electrons, right? So this is another way they've asked this. It was a hard question they asked on an old test. Take a look at this. So the key phrase there is polyunsaturated. If you see that, you want to think prostaglandins, okay? Okay guys, the next page that starts with triglycerides, the key thing I want you to know here is you can pause this and kind of skim it. They wanted you to know in a past test question about the idea that triglycerides are not part of the cell membrane, okay? The others listed on this page are, like phosphatidylcholine, phosphatidylanilamine, etc., inositol. Those are part of the cell membrane, whereas triglycerides are not. The other thing I would pay attention to, guys, is phosphatidylinositol. When you see that on the exam, you want to think arachidonic acid. As it says here in the bullet point in your notes, it's the primary source of arachidonic acid. So I want to show you the arachidonic acid pathway here for a second, okay? The way I always learn this in school is that you have membrane phospholipids, right? They get converted to arachidonic acid. Well, on the test, they're likely to be very specific. Which membrane phospholipid? It's this one right here. You guys know once you get to arachidonic acid, you can go down either arm of this pathway, and we'll talk about these more in just a little bit, but this is the big picture you want to know for now. The other thing I would say is the conversion of this phosphatidylinositol to arachidonic acid needs an enzyme. And that enzyme is called phospholipase A2. That's a big deal. And you probably remember that there is a drug that stops that enzyme. And that drug is called what, guys? It's steroids, yes, okay? That's what you want to pay attention to. Okay guys, let's go to sphingomyelins next. If you see that on the test, the first thing I want you to see is the second line in the notes. It says it's found in nerve cell membranes. It's an important part of myelin sheath, okay? If you read on at gangliosides, it says right there that sphingosine and N-acetyluramic acid, those two are found in gangliosides. I don't think either one of these are very high yield, to be honest. I wouldn't spend a lot of time on it, but those two things have been mentioned. The other thing I want to point out here is at the bottom of the page, guys, when we get into cholesterol, okay? You guys know at the core of cholesterol is going to be acetyl-CoA. Remember, that was one of the 4 things acetyl-CoA can do. You're going to remember again with acetyl-CoA, it cannot be used for gluconeogenesis. The other thing cholesterol needs besides acetyl-CoA right here in the notes is it needs NADPH, right? We've talked about that a few times. And you guys know that cholesterol is also the precursor for adrenal cortex hormones, okay? You remember the adrenal cortex hormones are salt, sugar, sex, right? That's also in the histology video, but pay attention to that. Look at this visual again to remind you salt, sugar, sex, and also the zones go, fast, Reggie, okay? So remember on the test, if they ask you about sugar, which is steroids, you want to be thinking fast. The fasciculata is what produces that, okay? Okay guys, on the next page when we talk about eicosanoids, you can see there the cyclooxygenase pathway and the lipooxygenase pathway. So let's show this visual again here of the arachidonic acid pathway. I showed it earlier to kind of build the base. Now let's go a little deeper. You can see cyclooxygenase is an enzyme that's going to produce prostaglandins, which are pro-inflammatory molecules, right? And make this note in your margin. If you forgot about this, make sure you remember prostaglandins protect your stomach. Okay, they protect your stomach. So that's why people can get ulcers who are on NSAIDs, because you know that NSAIDs block cyclooxygenase. Okay, so you can see that right there in the visual. The other thing is you also know steroids can cause stomach ulcers, right? Because steroids stop everything at the very top right here of the visual, right? So everything below that, including inflammation, including prostaglandins, which protect your stomach, are gone. So that's why you can get the ulcers, okay? So lipooxygenase, more as it says here in allergic reactions, they produce leukotrienes. Those are pretty prominent in asthma. Okay, that's how you want to associate that. Okay guys, one last point I want to make about the arachidonic acid pathways in the box. It's linoleic acid. Pay attention to that one, okay? You're going to see that again a few pages later from now, and you're going to see again that first of all, it's an omega-6, okay? So it's going to be pro-inflammatory. You guys can see right there, it's polyunsaturated. That is a huge point that they've mentioned several times on old test, okay? So pay attention to that. Linoleic acid, polyunsaturated omega-6 fatty acid. So guys, now we're going to get into lipid digestion. And you guys know, first off, triglycerides are what's going to be broken down. So when you think about this, first bullet point says when you have triglycerides in the duodenum, right, you're going to get a series of things that are going to happen. I want to show you this visual right now. And you guys remember that a triglyceride, has 3 chains, right? And so pancreatic lipase, right, once it senses this triglyceride in the duodenum, is going to be released from the pancreas, right? And it's going to break this down. And ultimately, you're going to get what's called here a mixed mycel, okay? And bottom line is with this mixed mycel, one of the things we're seeing a lot on old tests is, first of all, what is it? And you can see here in your notes, okay, the third bullet point, it says bile salts, free fatty acids, and monosaccharides combine to form this mixed mycel. So pause on that. You can skim your notes on it, but they are asking questions about mixed mycels. First of all, know that it's formed when a triglyceride is broken down. Know what it is right here. And then if you go to the next bullet point, the next dash, it says that hydrophobic interactions help to maintain the structure of mixed mycels. I know that's a lot of minutia. Maybe it's been a while since you've seen that, but that continues to get hit over and over. So pause on mixed mycels and really know the comments around that. Okay guys, as we get into the next topic, as you scroll down your notes, is going to be LDL, HDL. You're going to see this in the histology video as well. The bottom line is that we talk about is what's called the rule of 50s. Now I know not everything correlates with a 50 as you look down here because there's different sources, but the general rule of 50 applies really well. You want to start with the number 50 and say you want to be higher than 50. Take a look at this visual. When you think HDL, think higher than 50, but less than 100. So LDL needs to be less than 100. Triglycerides, think 3 times 50. So triglycerides are going to be 150. Total cholesterol is going to be 200. Okay, so when you think about those, again, I know the numbers vary a little bit. I don't think they're going to ask you the difference between 40 and 50 as a norm. I think you have to know general ballparks if they ask it. Make sure you pay attention to that. You also want to know underneath LDL. This is picky, okay? It says in our notes right here, once LDL reaches its target tissue, it's transported into the cell via receptor-mediated endocytosis, okay? That is how it works. So that's how LDL particles get into cells, is receptor-mediated endocytosis. So pay attention to that. You know also, by the way, LDL is considered lousy. It's the bad cholesterol. HDL is the good cholesterol. If they ask you on boards, why is HDL good? Because as it says here in the notes, it takes excess cholesterol from the organs back to the liver. Okay, that's how it works. So it's taking things to the liver. That's the key point you want to know about HDL. Okay guys, the next thing we want to talk about is xanthelasma. It's right there in the box. You guys know xanthelasma is going to be yellow. You know it's common in people with high cholesterol, but don't miss this. If they ask you the number 1 cause of xanthelasma, it's going to be aging. Okay, don't put high cholesterol as the number 1 cause. The next thing we're going to look at, everybody, is beta oxidation. As soon as you see that, I want you to think breakdown. Beta oxidation is the breakdown of fatty acids. Do you guys remember, by the way, what is at the core of a fatty acid? Do you remember? Well, you're going to start off with acetyl-CoA, right? And acetyl-CoA is going to get converted to malonyl-CoA. Does that ring a bell? So when you look at this, the first thing you want to think of is beta oxidation. Bam, this is so cool. Watch this. Every time you break down a fat in your body, right, you're getting acetyl-CoA, and as soon as you see acetyl-CoA, you're thinking, oh yeah, baby, I can put that in that diagram that we talked about. Acetyl-CoA, if it has that coenzyme A, right, can go into the Krebs cycle, right, it can have citrate synthase, all that good stuff. So think about it like every time you get acetyl-CoA, you're really close to getting into the Krebs cycle and producing energy, right? So that's a great thing about acetyl-CoA. The other thing is that you get NADH and FADH too. So take a look at this visual right here. Every time you do beta oxidation, which is again the breakdown of a fatty acid, you're getting these 3 things. So tell me that's not an awesome way to be able to produce ATP right off the bat. That's why fatty acids produce so much energy every time you break down a fat. Okay, that's a key thing I would pay attention to. When you think about acetyl-CoA, if you break down proteins, guess what you're going to get to? Acetyl-CoA. Carbohydrates, what are you going to get to? Acetyl-CoA. Lipids, what are you going to get to? Acetyl-CoA. So the take-home message is on the test, if you don't know the answer and acetyl-CoA is listed, pick it. Every time. With one exception. Do you remember what the exception is? Never put acetyl-CoA as the answer for being able to be used for gluconeogenesis. Okay? Okay guys, the next thing we're going to talk about is ketone bodies. And I want to show you this visual of a past test question we've seen many times. The key word in that question is the word starvation. If you see that on the test, immediately start thinking about ketone bodies. And we talked earlier about how ketone bodies can be used by the brain when there's low blood glucose, right? So pay attention to that. And if you look in the box, it talks about the 3 flavors of ketone bodies. And this is like a classic board question that shows up all the time. Acetoacetate, beta hydroxybutyrate, and acetone, right? If you keep reading in that box, you see the word fasting, right? So if you see fasting or starvation on board, start thinking ketone bodies and remember these 3 types of ketone bodies. Next thing we want to talk about, guys, is fatty acid synthesis. So I'm going to look there with you in the notes. You guys know that to produce a fatty acid, you need NADPH. Does that ring a bell? You're also going to need acetyl-CoA, right? That's what's going to be used to produce it. You know you're going to get acetyl-CoA converted to malonyl-CoA, right? And it needs this enzyme, pay attention to that, acetyl-CoA carboxylase, okay? That could be something to kind of pay attention to for a second. Every time you see malonyl-CoA, stop reading the question. You know it has to do with fatty acids. Okay, that's a key thing I would pay attention to. Okay guys, let's talk about nutrition. This is the next big topic we really want to nail. You can see there we talk about omega-3 and omega-6 fatty acids. So let me show you this visual to kind of compare between the two. You want to know the differences quickly. On omega-3 fatty acids, you can see we have alpha-linolenic. Think of it like you're lending something, lenic. Pay attention to that. You want omega-3, right? So think of it like you're lending someone more omega-3s because that's a tiny little detail because when you look at omega-6, it's going to be alpha-linoleic, okay, not lenic. So pay attention to those differences. But when you get to omega-3 again, your main examples here are going to be alpha-linolenic, EPA, and DHA, okay? So a lot of times as a doctor, I'll have patients come up to me and show me their fish oil tablets, right? And they're wanting us to see basically, is this a good fish oil or not? And what you're really looking for is how much omega-3s they have, right? Because a lot of the cheap fish oil are gonna have a lot of omega-6s, right? And the way you know that is to look at the omega-3s, which is again, the EPA and the DHA. All right, but as you get further, again, omega-3 are anti-inflammatory, omega-6 are pro-inflammatory, and as you look at the omega-6, it's going to be linoleic, right? Both LA and GLA and arachidonic acid. Okay, again, arachidonic acid, it's easy for you to remember. That's obviously promoting the production of prostaglandins, thromboxanes, leukotrienes, etc. So that's going to be all about inflammation. Now, in terms of a question, in terms of breakdown of fatty acids, you want to understand this mnemonic of 449. Okay, if you see this on the test, you get 4 calories per gram for proteins, 4 for carbs, and 9 for lipids. So when you break down lipids, you get more calories per gram. Okay, so the way they could ask this on a test is they could say something like, your patient has 8 grams of carbs, 8 grams of proteins, and 8 grams of lipid. You know, how many calories did they consume? Something like that. So if you know 449, right, you can do that math problem pretty easily. Think of it like 9 lips or something. If you want to look at this visual to help you remember, with lipids, it's all about 9 calories. The other, it's going to be 4. Okay guys, as we get into vitamins and minerals, you really want to know these well. This is one of my favorite parts of the lecture. We got to know the vitamins and minerals really, really well. And when you look at fat-soluble vitamins, you want to know DEAK. D, E, A, and K are going to be your fat-soluble. First thing with vitamin D, you want to think right here on the visual, think vitamin D is dangerous, okay? It's going to be your most toxic fat-soluble vitamin. Okay, and if they ask you on boards, where do you get vitamin D from? Well, you start with cholesterol, right? And sunlight will hit cholesterol and convert it to vitamin D, okay? And if they want to get really, really picky, right, the active form of vitamin D becomes active in the kidneys, okay? So if you want to go to your notes here for a second, you can see it says where you have all these 25 hydroxy vitamin D3 travels to the kidneys, where it's converted to the physiologically active hormone, etc. So I wouldn't memorize all those terms, but I would know the kidneys is where it gets transferred, okay? You guys know under function that it promotes hardening of bones and teeth, right? And you also know that a deficiency will lead to rickets in children and osteomalacia in adults, okay? So bottom line is I would know that, and the biggest concern with vitamin D, again, is it's dangerous if there's too much, and sarcoid, right, would be a good one I would pay attention to for this. So if I asked you this about right now, about sarcoid, is it more common in Caucasians or African Americans? You would say African Americans, right? You would want to know it's what age group? Middle-age African American females, okay? That's sarcoidosis. You want to know also that in this condition, it's idiopathic. We don't know why they get it most of the time, but in this scenario, obviously, if you have excessive vitamin D, it can lead to that problem, okay? Okay guys, now we're going to talk about vitamin E. Okay, one of the first things you want to know about this is that it's an antioxidant. So look at this visual here for a minute. When you see this, you want to be reminded that vitamins A, C, and E, ACE, right, and zinc are the main vitamins that make up antioxidants. Okay, so hone that in. That's really important. A lot of times patients will show me what they're on in terms of supplements, and I'm looking for those particular vitamins in terms of knowing which ones are going to be most helpful for antioxidant situations. Okay guys, the next topic we want to hit is vitamin A. And if they ask you on boards, what is the precursor for vitamin A? You want to be able to say right there in the box, beta carotene, yes, okay? And when I think of vitamin A, I think vitamin A is awesome, right? It's awesome for a couple different things. Number one, it's awesome for your enamel of your teeth, and that's a big test question they hit a lot. It's also awesome for epithelial tissue. That's why a lot of people will put vitamin A on their skin, because it promotes differentiation of epithelial tissue and causes it to be healthier. Okay, so let's look through the notes here together to make sure you got the key points. Underneath function, you'll see the first dash right there, maintains epithelial tissue, right? Next dash talks about bone growth and remodeling. Next one's all about enamel formation, right? Next bullet point's talking about rhodopsin, right? So you probably have heard before, if you have a lack of vitamin A, this is a leading cause of blindness, particularly night blindness. And it can also cause really bad dryness. And that's, again, the whole reason it causes dryness is because epithelial tissue, right, which produces glands like your lacrimal gland and stuff that produces fluid for your eye. If you don't have vitamin A, you don't get epithelial. Again, it's all about the epithelium regeneration. Okay, so that's the key points I would know about that. Don't forget about enamel. If you look in the box there, it says vitamin A and D. Do you remember this from earlier? We talked about how these are two examples, vitamin A and D, of why you can get hypoplasia of the enamel. Okay, so pay attention to that. The next thing we want to hit on is vitamin K. You know, as soon as you see vitamin K, you're thinking clotting. Vitamin K, it's all about blood clotting. Make sure you know vitamin A hates warfarin. They're enemies, okay? Warfarin is obviously a vitamin K antagonist. And when you think about that whole idea of blood clotting, make a note in your margin next to warfarin. Do you guys remember what platelets do? Platelets produce something. Does anything come to your mind right now when you think about that? You'll see this in histology as well. Platelets produce thromboplastin, okay? That's an enzyme that starts the whole cascade process, which eventually that process ends with fibrin forming the clot. Does that ring a bell? Good, okay, that's a big one. You guys can do this. You can get every one of these right, okay? That's a key thing I'd pay attention to. Okay guys, the next thing we're going to talk about is water-soluble vitamins, and vitamin B1 is a big one to know. This is a really good one that you'll likely get asked on. The first thing I think of with vitamin B1 is I think of thiamine, right? And I think alcoholics, okay? Because as you see here in the box, B1 is not stored in the body, so it can be depleted really, really quickly, and you need it in your diet. So alcoholics, if they go into an emergency room, they'll get injected, for example, with thiamine right off the bat to try and help rehabilitate them quickly. All right, so think vitamin B1, think thiamine, think alcoholics. And as it says right above the box, there's a condition, right, called Wernicke-Korsakoff syndrome caused by malnutrition in alcoholics. So pay attention to that. If you ever see Wernicke-Korsakoff, stop reading the question. It's all about alcoholism, okay? So that's a big one to know is vitamin B1. Okay guys, on the next page, you can kind of pause and skim that because those are low yield, but the next high yield item is going to be vitamin B12. So look at that, find that in your notes with me, and let's glance at it together. The big thing with vitamin B12 is all about pernicious anemia. Okay, so let me show you this visual for a second. Just as a reminder, it all starts with a parietal cell. Okay, you're going to see this again in histology as well, but a parietal cell is always going to produce two things. One is HCl right here in your diagram, the other one's intrinsic factor, okay? And intrinsic factor, as soon as you see that, I hope you're thinking, ding, 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 B12, because you need intrinsic factor to absorb B12, right? And the take-home message with all of this is that pernicious anemia is a disease that attacks parietal cells, okay? Once you attack a parietal cell, you don't get the production of intrinsic factor, so you end up with a major problem of vitamin B12, which, by the way, is this common? Yes, really common. Look in the box. The number one deficiency seen worldwide, really commonly associated with alcoholism, just like vitamin B1 deficiency that we talked about, okay? Last but not least, everybody, vitamin C. You guys know this is the same thing as ascorbic acid. When you see vitamin C, I want you to think C for connective tissue, right? We talked about earlier in the lecture how vitamin C is necessary for hydroxylation of proline and lysine residues, right? Do you remember that? Yes, okay, those are cofactors that cause hydroxylation and helps the collagen to be healthy. You also know, what are our antioxidants again, everybody? A, C, E, and zinc. Yes, okay, those are your main ones that you want to pay attention to. You know that if you don't have vitamin C, or a lack of that, can break down your collagen, you can end up with scurvy. Okay, that's a big one. And remember, the reason why we care about that in dentistry is obviously problems with bleeding of the gums and the teeth. The next one I want to hit everybody is folic acid. This is the next high yield one. This is the number one vitamin deficiency in the United States. You guys probably remember folic acid is used for making purines and pyrimidines, right? Hey, by the way, right above that where you have purines and pyrimidines, which is going to be used for DNA synthesis, do you remember what we talked about earlier is what you need? What compound do you need that donates electrons, right, for DNA synthesis? It was for ribose 5-phosphate. Anything come to mind? Yes, NADPH, right? So NADPH is going to be produced by the pentose phosphate shunt. NADPH donates its electrons, right, for fatty acids, steroids, and for ribose 5-phosphate, okay? So what we're saying here is folic acid is also needed for that process. You guys know the classic deficiency with folic acid is going to be spina bifida, okay? Okay guys, now we're going to talk about minerals, and as we look at this together, one of the things I want to point out right off the bat is this has not been high yield on past tests. So you can kind of pause the video and skim this if you want, but before you do that, go down to the bottom of the page and look at Wilson's disease. This is when you have too much copper. The first thing I want to point out about this is that there's another name for Wilson's disease in the box called hepatolenticular degeneration. Classically on standardized tests, they'll ask about hepatolenticular instead of using Wilson's disease, so be ready for either one. Bottom line is it's too much copper. I've seen case reports of these patients where literally they go crazy because they have so much copper in their system, and the doctor will finally figure out it's Wilson's disease, will treat them, and the difference is like night and day very quickly. So these are really inspiring patients to help if you ever get one. Next is fluoride, and I want to look at this with you guys. Look at the first sentence. It says it inhibits an enzyme, bacterial enolase. Do you think that's important? Yes, definitely highlight that. And I would also look at the next sentence, which it does a second thing. So I would know both of these things, which is that it converts hydroxyapatite to fluorapatite. And that's all about protecting what? Protecting enamel, yes. So I would know both of those two things and kind of look out for that on a test question. Okay, the next one we want to hit everybody is iodine. If you look here, you guys remember iodine's critical for thyroid function because it serves as a core component of T3 and T4, which is released by the thyroid. Just as a reminder, remember hyperthyroidism, you'll have high T3 and high T4, but you'll have low TSH. If you go on to iron, remember iron needs to be in its reduced or the ferrous state, right, to be used in hemoglobin. So pay attention to that. Okay guys, we're going to talk about zinc, and I want to summarize the key points up here on a visual for you. Number one, zinc is required for DNA synthesis, okay? That's a key point I would know. Number two, it's an antioxidant. That's another thing we've mentioned. And number three, and this is what I've seen most frequently on old tests, is that it's a cofactor for producing carbonic anhydrase. You might remember carbonic anhydrase is the enzyme used to make bicarbonate. That's in the box right there for you. Really star that to make sure you got the key points of zinc. That's the number one thing that continues to show up. Okay guys, on this next page where we talk about oxygen-free radicals, you can kind of pause and skim that page. The key thing to know is the antioxidants, which we've already talked about. And so if you look at that heading, you guys know antioxidants are responsible for eliminating free radical formation and thus protect cells, right, from oxidative, that's a key word to circle, stress. And if you look at the important antioxidants, glutathione is not one that we've mentioned much, but that's a key one. You also want to know vitamin A, C, E, and you can list zinc there as well. Okay, that's the key points you want to know about that. So guys, we've nailed kind of the key concepts in the vitamins and the minerals, so you can kind of sit back, relax, feel good about that.